Hello guys, welcome to Sqadia. This is Dr Danial Qasim. For more details about my profile, kindly visit sqadia.com. So today we're going to discuss about cell. We're going to discuss about its definition, its classification, and the organization of the cell. Plus we're going to be discussing the comparison of the animal cell with the precellular forms of life, plus the physical structure of the cell, the organelles, the membrane bounded organelles, and the cytoskeleton of the cell, plus the functional systems of the cell and the locomotion of the cells. In the end, we're going to just summarize the whole lecture. So what is cell? Cell is the smallest and functional unit of an organism, typically microscopic and consisting of cytoplasm and a nucleus enclosed in a membrane. Microscopic organisms typically consist of a single cell, which is either eukaryotic or prokaryotic. So this is the cell. You can see it is bounded by cell membrane. It's a nucleus, which is the control center of the cell, which is contiguous with the endoplasmic reticulum. Plus we have flattened sac network of Golgi body, and we have lysosomes and mitochondria, which is the powerhouse of the cell. So cell, there are 75 to 100 trillion of cells. Some are specialized cells. They form organs and the organic system of the human body. Their function is to produce energy, regeneration of new cells, and the constituents of the extracellular fluid can diffuse back and forth in the cell and in the extracellular fluid. So cells can be classified into two types. One is eukaryotic and the other one is prokaryotic. Eukaryotic is a type of cell that does have its chromosomes surrounded by a nuclear membrane, and it possesses a nucleus. So replication of cells and generation of new cells is through the process by the control center of the cell, that is the nucleus. It is bounded, you can see the diagram, it is bounded by the cell membrane. There is a nucleus with a nucleoli, and the ribosomes, endoplasmic reticulum, some specialized organelles are also present, like mitochondria, ribosomes. And in prokaryotic cell, the type of cell that does not have its chromosomes, and it is surrounded by a nuclear membrane. So it means it doesn't have a nucleus. You can see in this diagram, which is the bacteria, viruses, these are type of prokaryotic cells. So in this type of prokaryotic cell, we have a cell wall. You can see the diagram of a prokaryote with a nucleoid in the center with the ribosomes. You can see the capsule, which is a protective covering of the cell, and plus for it is surrounded, the capsule is surrounded by the cell wall, and then that cell wall is surrounded by the cell membrane, and you can see the flagellum is present in it. So human cell or the animal cell and plant cells are the examples of eukaryote, and bacteria or rickettsia, viruses, are the example of prokaryote. We're going to be discussing the difference between the two forms, and we are going to find out few distinctive points between the two in further slides. So how the organization of cell comes into being. So first is hydrogen atoms or oxygen atoms. The atom, the smallest particle or the smallest constituent of life is atom, which fuse to form or combine to form molecules or a compound. Like for example, if a hydrogen atom combines with the oxygen atom, so we're going to get the water molecule, and that molecule combine to form organelle, like for example sarcoplasmic reticulum in smooth muscle cell. So a variety of molecules combine to form fluid and organelles of the body cell. These organelles then fuse to form a cell, a smooth muscle cell or a red blood cell, white blood cells. So these are all type or forms of cells, specialized cells. These cells then combine at the tissue level and they form a tissue. Then this tissue and tissue is formed by the community of similar cells, like for example a smooth muscle cell or two or more smooth muscle cells can combine to form a smooth muscle tissue, and white blood cells combine to form a white blood cell. So it's a process, or it's a phenomena in which the cells they combine to form tissue, and tissue combine to form organs, and then organs working together or working with other organs, they form an organic system, and hence organism is formed. Since you can see the levels, we are starting from the atom level, and then there's a molecule level, then we jump to the cellular level, and this cellular level then jump to the tissue level by the combination of the similar varieties of cells. Then there is an organ level in which different organs working together to form an organic system, an organ system, like for example a respiratory system in collaboration with the heart, in collaboration with the with kidneys and with urinary system. So they're all working together, and that's how the body mechanic works, the body mechanic functions. So what is a protoplasm? Protoplasm, the substances that make up the cell, collectively called as protoplasm. So it is composed of water, which is 70 to 85%, electrolytes, which are inorganic chemicals for cellular reactions, namely potassium, magnesium, phosphate, sulfate, bicarbonates, and small quantities of sodium chloride and calcium. Proteins are 10 to 20%, which are structural and globular proteins. Lipids 2%, with cholesterol, phospholipids, triglycerides, and neutral fats, and carbohydrates almost forming 1% of the total cell mass, and they can be high as 3% in muscle mass and 6% in liver cells, because glucose is taken up in the liver for the process of glucose formation or for utilization of glycolysis. So now we are going to compare the animal cell with the precellular forms of life. So we discuss about the eukaryote and the prokaryotic cell. We know that the eukaryotic cell is the animal cell or the plant cell. We know it has the nucleus, and we know that prokaryote doesn't have the nucleus. It is surrounded, the prokaryotic cell is surrounded by the capsule, by the cell wall. However, there is absence of cell wall in the animal cell. For the precellular forms of life, like bacteria, like viruses, they are 1,000 times smaller, or 1,000 times smaller in diameter than the animal cell. However, their volume is 1 billion times 1 billion times lesser than the animal cell. The anatomical organization of viruses, of small viruses, or the prokaryotic cells, they are very complex. So essential life giving constituent of these small viruses is nucleic acid, like we discussed before, and which is embedded in the core of protein, which is composed of DNA and RNA. There are specialized protein chemicals that are present that become the integral part of the cell, and they perform their specific functions. They survive generation after generation, and they get evolved. In Rickettsia and in bacterium, there are special organelles that start to develop in the later part of their life, and they perform specialized functions for the cell. So and we know, so that is the main key points that are present in the precellular forms of life, and that's how we can compare with the animal cell. We know that animal cell has a nucleus, a control center, and have organelles. They perform their specialized functions. So each new cell of the animal cell is similar or resembles its progenitor. So animal cell, you can see the diagram, the animal cell and the precellular forms. We have small viruses, rickettsia, and the bacterium. You can see the small virus is 15 nanometers, and 350 nanometers is rickettsia. Bacterium is 1 micrometer, and you can see the cell which is 5 to 10 micrometers. So it is almost 1,000 times greater in diameter than the small virus. The volume is also 1 billion times greater than the small viruses. So we know that they have the main difference between the two. They both perform their specialized functions. They both have their unique capability of performing different functions in their own environment. So far we have discussed about the cell, its definition. We have discussed the classification and the organization of the cell, and we have compared the animal cell with the precellular forms of life. Now we're going to discuss the physical structure of the cell, and going to discuss the detail about their function and the function systems of the cell with the locomotion of the cells. Now, the physical structure of the cell, it is a bag of fluid containing highly organized physical structures known as organelles, which are present in membrane bounded organelles, which are cell membrane, nucleus, Golgi apparatus, lysosomes, and mitochondria. And there are some cytoskeleton. Cytoskeleton possesses the microtubules, microfilaments, and there are intermediate filaments that are present inside in the cell, which are very important, and they perform their normal function. So we know the cell is surrounded by a cell membrane, which is composed of protein and lipids. It provides a barrier to prevent free water flowing and water soluble movement from one cell to another. There is a free movement of protein, and there are pathways for the substances to enter in and move out into the extracellular fluid. So all the organelles, they are placed in a clear fluid inside the cell that we call as cytoplasm, and it is surrounded by the cell membrane, and it contains all the cell organelles. Now here's a diagram of a cell. We can see all the organelles, plus we can appreciate the cell membrane. You can see the Golgi apparatus, there's a nucleus. They all are membrane bounded organelles: the endoplasmic reticulum, the nucleus, the Golgi apparatus, the mitochondria. We have lysosomes and peroxisomes. We have intermediate filaments. Some centrosomes are present with the microtubules, and there's a chromatin material you can see inside the nucleus, which contains the DNA and RNA, and rough endoplasmic reticulum, which is studied with the ribosomes. So all will be discussed in detail in the following slides on the following presentation. Now the cell membrane, also known as the plasma membrane or the cytoplasmic membrane, is a biological membrane that separates the interior of the cell from the outside environment. Lipid bilayer, it is made up of lipid bilayer with inserted proteins, which we call as glycoproteins. So lipid bilayer is composed of phospholipids and cholesterol. Phospholipids are water soluble portion has a water soluble portion and the fat soluble portion. So water soluble portion, which we call as the hydrophilic portion, and the fat soluble portion is the hydrophobic portion. So hydrophobic portion forms the tail of the lipid bilayer, which is facing towards each other, and the hydrophilic portion forms the head containing two surfaces of the membrane with the surrounding interstitial fluid. Lipid bilayer membrane is a floating membrane which is highly permeable to lipid soluble substances like oxygen, carbon dioxide, and alcohol, and impermeable to ions and glucose. There are two types of membrane proteins: integral proteins and the peripheral proteins. Integral proteins, they protrude through the membrane. They have pores through which the ions can diffuse, and they are also called as the carrier proteins, or they contain the carrier proteins. The peripheral proteins attached with the integral proteins act as an enzymes, catalyze the chemical reactions of the cell, and does not penetrate. Glycolipids and glucoproteins protrude outside of the cell. Proteoglycans loosely attached to the outer surface. Entire surface is covered by the carbohydrate coat called as glycocalyx. They repel the negative charge molecules. Attachment with the other cells act as receptors for binding hormones. Now you can see there's a cell membrane structure, which is the lipid bilayer. You can witness the hydrophilic ends and the hydrophobic ends, which are the head and forms the tail. So these are the hydrophilic heads, and these are the tails, which are the hydrophobic tails, which are lipid soluble, and this is the head is the water soluble component of the cell membrane. It has the fluidity in its shape inside the cell. It creates the fluidity for the cell. You can appreciate the proteins. The protein channels, they are the globular proteins, which are present the glycoproteins, and you can see the integral proteins, which is protruding to the cell membrane. Then there we have the peripheral protein, which does not protrude and remains inside the cell, and it also the protruding or the globular integral protein. It is also a mode or a transport protein, a channel. It provides a channel, like we can see over here. So this is all in all, that is a picture of a fluid mosaic model, a lipid bilayer membrane having hydrophilic and the hydrophobic ends, and we know the tails, which are the hydrophobic ends, they facing towards each other, while the heads, which are the hydrophilic heads, they face the extracellular fluid or the interstitial fluid of the cell. So endoplasmic reticulum, which is a membrane-bound organelle present in the eukaryotic cell, they form the interconnection network of flattened membrane enclosed sacs or tube-like structures known as cisternae. So they perform a network, or they create a network. They are interlinked throughout the cell, and they are of two types. One is rough endoplasmic reticulum, and the other one is smooth endoplasmic reticulum. Rough endoplasmic reticulum is convoluted, flattened, sealed sacs. They are contiguous with the nuclear membrane, and they are studded with ribosomes, and they are involved in the protein synthesis. While smooth endoplasmic reticulum is a tubular network that is present throughout the cell. They are the lipid manufacturers and its associated products. So it is endoplasmic reticulum. It is a membrane-bounded organelle that performs a synthesis of proteins and lipid synthesis and its associated products. So they are associated with the messenger RNA in synthesizing many proteins, which enter the Golgi apparatus and for the further modification of the molecules to be utilized inside the cell or outside of the cell, which are to be released. Now this is a picture of endoplasmic reticulum. This is smooth endoplasmic. So we can see the diagram of endoplasmic reticulum. We have smooth endoplasmic reticulum, which is not studded with ribosomes. So these are the small molecules you can see, you can find on the surface of the endoplasmic reticulum, which we call as rough endoplasmic reticulum. These are the ribosomes. And you can see the outside membrane or the outer membrane of the nucleus is in continuation with the membrane of the rough endoplasmic reticulum. And so these are the spaces, the network of rough endoplasmic reticulum which is interconnected with each other. There are spaces which we call as cisternal spaces. They're forming a network which is called as cisternae, which we have discussed. And there's a spaces between these cisternae and that are called as cisternal spaces. Golgi apparatus. We know that after the synthesis of proteins in the endoplasmic reticulum, for further modification, it is transferred into another network of membrane-bounded sacs that are present inside the cell, which we call as Golgi bodies or the Golgi apparatus. So they are also named as dictyosomes. They are the flattened membrane enclosed discs having cisternae network, which is called as cis-GN or cis-Golgi network. Then we have the final cisternal network from where the proteins are packed and secretory vesicles are released. It's known as trans-GN or trans-Golgi network. So the vesicle which is transported to the Golgi apparatus, these are the flattened network of Golgi apparatus, which is the cis-GN having the cisternae of the flattened membrane sacs. You can see the cisternae, they are interconnected. So the vesicles, when they come from the endoplasmic reticulum, when they are transported with the help of the messenger RNA, they enter the cis phase or fuse with the cis phase for further modification. Once the final product is formed, it is transferred to the trans phase or the trans-GN network of the Golgi apparatus, from where the secretory or the outgoing transport vesicles are generated after the modification of various molecules that are coming right from the endoplasmic reticulum. Now the lysosomes. Lysosomes are small vesicles that are surrounded by the membrane containing digestive hydrolytic enzymes. They destroy the unwanted substances, the food particles. Lysosomes is involved in various processes, cell processes including secretion, plasma membrane repair, and destroying of the foreign objects, and they are involved in the exocytosis of the cell. So what happens that any damaged organelle or the foreign particle that enters the cell or that needs to be destroyed, or some repair at the cell membrane is required, it fuses, that particle or that substance fuses with the lysosomes, forming the endosome. So it breaks down or degenerates or hydrolyzes performing an enzymatic reactions within the lysosome to break down that product, to break down that foreign object, and they are to be released, they are excreted outside of the cell. So lysosomes perform a specialized function in maintaining the internal environment of the cell healthy in the stable and in the stable form. So it is kind of a security check or a security, you can take lysosome as a protective organelle from providing a protection against different foreign objects that enter the cell and causing the damage to the other parts of the cell. So having its digestive enzymatic capability, it helps in maintaining the environment of the cell. Mitochondria. So mitochondria is a double membrane bound organelle which is present in all of the eukaryotic organisms. They are found in some in multicellular organisms. However, for example, they are present in all the eukaryotic cell, but they are absent in some of the more specialized cell, like the red blood cells. A mitochondria contains outer and inner membrane, which composed of phospholipid bilayers and proteins. The two membranes have different properties. Because of this double membrane organization, there are 5 distinct parts to the mitochondria. There is an outer mitochondrial membrane, there's intermembrane space, that is the space that is present between the outer membrane and the inner membrane. Then we have the inner mitochondrial membrane, and we have the crystallized space formed by the enfolding of the inner membrane, and then we have the matrix, which is the space inside the inner membrane. So the hydrogen and carbon dioxide is removed in a process called citric acid cycle. ATP is generated through mitochondria, utilized in other cellular functions, and it has the self-replicative process. It has own DNA. So mitochondria, we have the outer membrane, and we have the space between the outer membrane and the inner membrane. These are the crystali. You can see the enfoldings of the inner membrane, and this is the matrix from where this Krebs cycle or the citric acid cycle does occur, and ATP is generated with the ATP synthase particles that are the enzymes that provide the formation, or they function as providing a catalyzing capability in converting ADP to ATP. So they are the ATP generated organelle, and having its own DNA, so it has the self-replicative process. So nucleus in cell biology, the nucleus is a membrane that is enclosed that encloses each organelle found in the eukaryotic cells. It is the control center of the cell, having the nuclear membrane, which is composed of 2 membranous layers. The outer layer is continuous with the endoplasmic reticulum, and there is also space between the 2 layers. Both layers have numerous pores, almost 100 nanometers in diameter. The largest structure that is present in the nucleus is nuclei, which is made up of proteins, DNA and RNA. Ribosomal RNA is stored in nucleolus and transported through pores to outside of the cell, forming mature ribosomes. So enzymes in the cytoplasm, reproduction, and the characteristic of the protein, these are the functions which are performed by the nucleus. So nucleus, you can see in the diagram, there is a nuclear envelope, or the outside. There are 2 membranes, there's the outside membrane and the inside membrane, with the numerous pores, and the outside membrane of the nucleus is continuous with the rough endoplasmic reticulum, which is studded with the ribosomes, which we have discussed earlier about the function of rough endoplasmic reticulum. So we have the chromatin material, which contains the DNA or RNA, having the capability of reproduction. So chromosomal, all the mitosis, the process of mitosis, meiosis, that does occur in the control center of the cell. Now the cytoskeleton. The cytoplasm is the material that is present within the living cell, excluding the cell nucleus. It comprises cytosol, which is a gel-like substance enclosed within the cell membrane, and it doesn't contain the organelles. And the organelles, which are the cell's internal substructures, and the microfilaments and microtubules forming the cytoskeleton of the cell. There are sometimes present few intermediate filaments that are also present, that provides an attachment of the cells with the different parts of the cells, which we're going to discuss later. So microfilament, the first part of the cytoskeleton. Microfilaments are the actin polymers, which are the actin filaments. They are 7 nanometer in diameter with 2 strands of actin, tough and flexible network. They provide cell motility, contractile movements, cytokinesis, mechanical stability, endocytosis, and exocytosis. Microtubules, these are 25 nanometer in diameter. It is an important part of a cytoskeleton, polymerization of globular and alpha and beta proteins. It performs intercellular transport, make up cilia and flagella, separation of mitotic spindles, movement of secretory vesicles and organelles, and intercellular macromolecules. So this is the structure of a microtubule. You can see the polymerization of alpha tubulin and beta tubulin, which are coated with red and yellow. This is a cross section, and you can see the hollow structure inside. These are the microfilaments, microfilament based movements. You can see which are the actin filaments. So the head of the myosin, when attaches with the actin filament, it causes the movement, causes the contraction, which is very important phenomena in the providing an action potential in the after the action potential of the skeletal muscle. There is a contraction coupling that does occur in the skeletal muscle, and it causes the contraction of skeletal muscle and the voluntary movement. So it is an important element of the cytoskeleton, each performing their normal regular function. You can see the microtubules. They perform the intercellular transport. They make up the cilia and flagella with the secretion of with the separation of the mitotic spindles. So microtubules, they are very important at the chromatin level. During the process of anaphase, the mitotic spindles that are present at the center of the centromere, they help in providing a separation point, or a separation of the two mitotic spindles, or a separation of the two chromosomes to their respective poles. So they provide a point of separation between the two chromosomes in the mitotic spindles at anaphase of the mitosis. So movement of the secretory vesicles, all the secretory vesicles that are coming from the endoplasmic reticulum to the Golgi apparatus, from the Golgi apparatus, the packed vesicles, then they are moving, they are ready to be moved outside of the cell. They provide this motion of the cells and the few intercellular movements of the organelles and the intracellular macromolecules like oxygen coming inside of the mitochondria. All these movements of the diffusion, they are affiliated, or they are associated. They help in functioning these movements and performing the specific function for each of the organelle. Now, intermediate filaments. Intermediate filaments are the important part of the cytoskeleton. The main feature or the main point of the intermediate filament is that it provides an attachment point, like desmosomes, cell adhesion. It provides a cell adhesion to different cells between the cell matrix and cell, between a cell of one tissue with the cell of the other tissue, or between the two similar cells. So it provides an attachment, junctional points. So there are two or three types of desmosomes, hemidesmosomes. There are junctions, junctional points, or the junctional epithelium that providing the cell adhesion between the different cells. So the diameter of the intermediate filament is 10 nanometers. There are 6 types of intermediate filaments. Number 1, type 1 and type 2 is acidic and basic keratin. Example are epithelial cell and hair keratins. Type 3 is desmin, vimentin, GFAP, and peripherin in neurons, in astrocytes, leukocytes, and endothelial cells. Then we have type 4 and the axon of neurons, which are the neurofilaments. Type 5, which are the nuclear lamins, and type 6, which is the nestin. Now, the function system of the cells, there's an ingestion of the cell through endocytosis. You can see these are the proteins or the contractile proteins which are covering the which are coating the pit of the cell membrane having the receptors. So any molecule that becomes attached with the receptors, they perform a vesicle. They contract. There's a contractile element that is coating the cell membrane surface. It performs its function forming a vesicle, which is a coated pinocytic vesicle. This vesicle then it's presented with the endosome. There are hydrolytic enzymes that are present within it. They perform their normal function. There's a digestion in the lysosome. So, and the respected or the vesicle which was once coated with the contractile, which was coated with the fibrillar protein or the coated protein, which is the contractile filament glycerin that is present surrounding the pinocytic vesicle, moves back to the inner surface of the cell membrane, and the vesicle containing the receptors move back to the cell membrane as well. So this is the function of the cell, that is the ingestion of the cell that is performed during the endocytosis. Now we have phagocytosis, and in phagocytosis there is a pseudopodia that surrounds the once microbes becomes attached to the cell membrane. The pseudopodia perform their function. They move outside. Performing the pseudopodia, finger-like projections are created on the outside of the cell membrane. The microbes are engulfed inside the cell. The vacuole containing microbe forms, and you can see this is a vacuole which containing the microbe. They become attached with the lysosomes. They're performing the hydrolytic enzyme capability, and there is all the toxic compounds and the lysosomal enzymes destroy microbes, and microbial debris is released by the exocytosis through the you can appreciate the exocytosis. All the remnants of the microbes are excreted outside of the cell. So that's how the phagocytosis works. So phagocytosis, always remember that there's a difference between the pinocytosis and the phagocytosis. In pinocytosis, which we have just discussed earlier, it is for the small globules or for the small molecules that are present in the extracellular fluid that needs to be digested within the lysosomes. So that follow this process. While the phagocytosis, the large products or the large particles such as the bacteria and the degenerating tissue, which are the larger in diameter than the smaller molecules, they go through a process which is called as phagocytosis by the process of pseudopodia that's surrounding the cell, forming a vesicle containing the microbes. They are presented to the endosomes and they perform their digestive function, and the waste products are then excreted outside of the cell. Now other function system of the cells are the synthesis of the cellular structure. Like for example, in endoplasmic reticulum, the ribosomes are attached, which we have discussed, and they perform the protein function. There are congregation with the carbohydrates forming the glycoproteins. So proteins are cross-linked, folded, and shortened to come to more compact molecules. You can see here this is a polypeptide molecule having the glycoprotein with the glucose portion and the protein portion. This sugar component then transported through the transport vesicle. They are budded off from the endoplasmic reticulum and they are presented to the Golgi apparatus for the further modification. So that's how they work, and smooth endoplasmic reticulum will form lipids like phospholipids, cholesterol, that are transported to the Golgi apparatus for the further modification. So that's how the synthesis of the proteins from the endoplasmic reticulum, they are transported to the Golgi apparatus to perform for the further modification. And with the help of the messenger RNA, the transport vesicles are shifted or transported to the Golgi apparatus for the further modification. That's how it does work. That this is a protein processing and secretion. You can see the ribosomals. This is the cytoplasm. So ribosomals assemble proteins from polypeptides entering the rough endoplasmic reticulum. Proteins move through the rough endoplasmic reticulum where they are further modified. Transport vesicles containing the proteins are pinched off from the rough endoplasmic reticulum, and they then move towards the Golgi apparatus. Now this is the cisGN, then it is transGN. So cisGNA modified, then it transported to the transGN for the further modification, and then it moves towards the to the inner surface of the cell membrane, from where they are excreted. So they are secretion of proteins from with the help of the exocytosis. So this is the process how the synthesis of the proteins worked. We have the glycoproteins which are transported to the Golgi apparatus, the phospholipids, the cholesterol, they all are transported from the endoplasmic reticulum network to the Golgi apparatus for the modification, and then the secretion of proteins or the secretion of lipids does occur with the help of this process. So anything, any nutrition we eat, we take carbohydrate, protein and lipids, they all are broken down into glucose, amino acids and fatty acids. So ATP is generated in the mitochondria, which is a nucleotide composed of nitrogenous base, pentose sugar and 3 phosphate radicals. These are the high energy phosphate bonds that are present between the last 2 phosphate radicals and the remainder of the molecule. 5% of the ATP is generated with the help of the glycolysis that occurs in the cytoplasm through the formation of pyruvic acid. So this pyruvic acid enters the mitochondria and is converted to the acetyl-CoA enzyme, which is an important product of the electron transport chain, and hydrogen ions are created, which provides an energy, and ADP is converted to ATP and energy is generated. So ATP synthase is an enzyme which is a large globular protein that helps in the formation of the ATP, converting ADP to ATP. You can see in this diagram the ATP is converted to ATP after the pyruvate enters the mitochondrial matrix, and it is converted to acetyl-CoA with the help of the citric acid cycle. ATP is converted to ATP and hydrogen is moved back into the pool and fuses with the oxygen to provide energy. So newly formed ATP moves out of the mitochondria to perform the energy to the various parts of the cell. It has chemoosmotic mechanism. It is involved in the membrane transport, in the sodium potassium pump, perform the contractile movements and the ciliary movements and the synthesis of the chemical compounds. Now the locomotion of cells. We have 2 types of locomotion. One is amoeboid and the other one is ciliary movement. So important type of movement that occurs inside the body, like for example specialized muscle cells in the skeletal, in the cardiac and the smooth muscle, constitutes almost 50% of the movement of the cells. So there are 2 types of the cells, which are amoeboid and ciliary movement. Amoeboid locomotion that is present in the white blood cell. There is a pseudopodium protrusion by the continual exocytosis at the leading edge and endocytosis at the mid portion or at the rear portions of the cell. One effect is pseudopodium attaches with the fixed tissue. The whole cell then moves in the forward direction with the help of receptor protein that lines the exocytotic vesicles. Second effect is by the filamentous network of actin and myosin binding. Energy is driven by ATP, and there is a chemotactic substances initiate amoeboid movements. So next movement is the ciliary movement, which is a whip-like movement of the surface of the cell with the help of ATP and magnesium and calcium. In respiratory airways and uterine tubes layer of mucus to move towards the pharynx. That moves towards the pharynx at once, clearing the passageway. In uterine tubes, a slow movement of the fluid from ostium to the uterine cavity. This fluid transports ovum, from ovary to the uterus. So there is a mistake in the slide, is that then there is respiratory airways, and there's a movement of mucus that moves towards the pharynx at 1 centimeter per minute, clearing the air passageway. And uterine tube layers, they perform their separate function, and which are the slow movement of the fluid from the ostium to the uterine cavity. This fluid transports the ovum from the ovary to the uterus. So amoeboid movement is the movement which we have discussed with the help of projecting the pseudopodium. We have the exocytosis that occurs at the leading edge. So exocytosis occurs here, and endocytosis occurring at the rear portion, pushing the movement or moving the cell forward, or by creating a pseudopodium, a finger-like projection to the receptors or to the substances that needs to be get engulfed and performing the exocytosis. So it moves in the forward motion like that with the help of the pseudopodium, with the help of the chemotactic substances. And sometimes with the help of ATP generation, by the help of cross-linking or cross-bridging of the actin and the myosin filaments, it causes the movement of the cell in the forward direction. Then we have the ciliary movements. They have two strokes. One is the effective stroke and the other one is the recovery stroke. That is calcium and the magnesium are the important aspects or the important constituents in performing the ciliary movements. So beating of the cilia, that usually occurs in two phases, that is the power or the effective phase and the recovery phase. During the power phase, the cilia become straight and stiff and move against the medium such as water. This movement pushes water backwards and propels the organism forwards. In the recovery phase, the cilia becomes limp and return to their original position in the curved state, offering a minimum resistance. So this is the recovery stage. You can see the curving of the cilia. This is the recovery stage of the process, and they perform the minimum resistance. As we know that here it is performing a whip-like motion and it is propelling the organism in the forward direction. So it pushes it's like swimming, having a butterfly stroke. When we do swimming, we push the forward, we push the water backwards, and our whole body is pushed forward. So the same process applies over here in the power stroke of a cilia. The cilia is stroked in the forward direction, pushing the water backwards, and the organism moves in the forward direction. So we're going to summarize the whole lecture. What we have studied till now is that we have studied about the cell, its definition. We came to this discussion about the difference between the animal and the precellular forms of life. We have discussed that the small viruses or the bacteria, they are smaller in diameter than the animal cell, and we know it is a part of prokaryotic family, and animal cell is a part of eukaryotic family, having a nucleus, while the prokaryotic cells doesn't possess nucleus, although they have the nuclear material. Then we discussed about the different membrane bounded organelles. We have discussed about nucleus. We have discussed about mitochondria, ribosomes, endoplasmic reticulum, the Golgi Apparatus. We have discussed each organelle and its associated function. Then we studied about the phagocytosis, the pinocytosis. Pinocytosis is a process of in a cell which ingests the small particles or the small molecules which are then digested by the lysosomes, and then they are excreted outside of the cell. Then we studied about the phagocytosis, the microbes, the larger particles or the larger foreign objects. They are engulfed inside the cell with the process of phagocytosis, and they are presented to the endosomes, and their waste products or their wastes are excreted outside of the cell. With the help of the exocytosis, we have discussed about the energy that is formed inside the cell, inside the mitochondrial matrix, by the ATP generation, by the conversion of ADP to ATP with the help of glycolysis. The pyruvate that is formed in the cytoplasm move towards inside the matrix, forming acetyl-CoA, and then the citric acid cycle. With the help of citric acid cycle, ATP is generated and energy is formed. So this is what we discussed today. We have checked all the topics that were presented to you today in this lecture. I hope you liked it. Thank you for watching sqadia.com.